Everybody, my name is Anna Neubeck and I'm an associate professor in geochemistry at Uppsala University. And as Maria Paz already have said, I'm working with mineral microbe interactions mostly, so not directly with active hydrothermal systems. So in my talk, I will go through, the first part will be quite basic on the hydrothermal vents and hydrothermal systems in general. And then the second part of my talk is going to be a little bit more specific on what I'm doing and what I'm working with at the moment about my research on mineral microbe interactions. Let's see. Yes, so hydrothermal vents, what are they? As you can hear on the already in the word hydro and thermal, it's about water and heated water, and the hydrothermal system is the interaction between rocks and circulating hot water. So the contact between rocks and water, basically. And there are two types, a terrestrial type, which includes geysers, fumaroles, which are vents from which hot gases and vapors emits, and hot springs and active serpentinization areas. And in the ocean environment, you have smokers, but black smokers, white smokers, and carbonate-rich smokers. And the hydrothermal systems are distributed all around the world, mostly located on plate boundaries, as you can see here. And there are thousands of hydrothermal vents all over the world, and there are yet a lot more to discover. So the major energy sources for hydrothermal vent systems, so the major energy source for heating the water is either through the vicinity to a hot magma chamber or through the serpentinization reaction. And serpentinization is an exothermic chemical reaction in which olivine, which is silica depleted iron-rich mineral, is reacting with water, producing the hydrated version of olivine, which is called serpentine, and some other minerals such as brucite and magnetite. And it's also producing hydrogen gas, but most importantly is producing heat. And the heat that is produced in this serpentinization reaction is directly proportional to the amount of water taken up to form the serpentine minerals. So for every cubic meter of rock that is olivine that is consumed, it's consuming approximately 300 kilograms of water. And that in turn is producing approximately 660 million joules of heat. And in order to understand how much that is, you can make a comparison. So for example, to charge a smartphone, you need 3,600 joules. And for one lightning strike, it's approximately 10 billion joules. So a cubic meter of olivine rock that is altered by water is producing quite a lot of energy and quite a lot of heat. And if one looks at the actual amount of heat that is produced, serpentinization is capable of raising rock temperature to about 260 degrees Celsius. And that is if we do not include the transport of heat or include the cooling of the rocks itself. So it's a reaction that is quite powerful, and that has been suggested to be one of the driving forces for some of the hydrothermal vent systems on the ocean floor, even though that is a bit disputed. But so before I talk about life in hydrothermal vent systems, I was going to talk about the history of hydrothermal vent discovery, because it's a fantastic story of scientific discoveries. So in 1880, the Russian ship Vitas was sampling the waters outside Mecca in the Red Sea. And they took up water samples from 600 meters depth, which is approximately 2,000 feet. And they discovered that these waters were warmer and saltier than the surrounding waters. And they had no idea why this was. And if you can see in this picture here, they were sampling from one of these 3 pockets here outside Mecca. And these pockets are, as you can see here in the seismic reflection image, small depressions in the ocean, or small, they're quite big. And you can see here in the seismic reflection image that they're filled with salt brine, so very heavy salty water in these, filling up these pockets. And this seismic reflection image, it's not from the 1880s, of course, but it shows where they were sampling from. So they took up these samples, and they could feel that these samples, the waters were very warm. And that sparked some interest in the research community in general. So many countries followed this expedition. So Austria in 1897, Germany in 1898, Sweden 50 years later in 1947, and US and Britain just a couple of years later, to also sample these mysterious salty waters of the Red Sea. And every one of them could also notice that these waters were hot and salty. And one of the early explanations for this was that the equatorial sun heated the waters, evaporating the water and increased the salinity. However, this couldn't explain why the waters were so hot. So this sparked even more interest to go and sample these mysterious waters. And not only in the Red Sea, but in other places where they had an active plate boundary. So in 1965, Woods Hole Oceanographic Institution passed through the Red Sea. They were going to the Indian Ocean to sample other things, but they couldn't resist sampling also from the Red Sea because of the hot waters. So they took up some sediment samples and some water samples, and they recorded a temperature of 56 degrees. And they were surprised by this very hot temperature of these waters, because it was hotter than previously recorded. And they also looked at the sediments that they were taking up, and they could see that these sediments were full of shiny metals and full of unusual colors. And they were, well, they could see these fantastic metals that were precipitating out in these sediments. So then after this expedition, there were some extensive sampling of the ocean seafloor in the 1960s and 1970s. And a lot of samples were collected, and all the samples, they could conclude that these sampled rocks were hydrothermally altered. So they could see that the rocks themselves had been altered by hot water. And the scientists started to connect the dots when they were looking at the rocks from Cyprus. And they compared these Cyprus rocks with the sampled rocks from the Red Sea. They could see that they were very, very similar. And they started to realize that these deposits, which were very metal-rich ores in Cyprus, maybe was an old hydrothermally altered piece of oceanic crust that were on land. So they thought that this Cyprus suit of minerals were these ophiolite deposits that were old oceanic rocks. And then ophiolite is a sequence looking like this here on the right. You have sediments on the top, and then pillow lavas directly under that, and then dikes, gabbros, and ultramafic rocks below. And this was what was also discovered in Cyprus. So this started the search for hydrothermal vents even more, and several expeditions was launched to measure the water temperature and small earthquakes. And in 1977, a large expedition went to the Galapagos Rift, and they were measuring the temperature over the rift, and they could see that at some point there was a spike in temperature, and this spike lasted for approximately 3 minutes. And at the same time, they could also discover micro earthquakes as connected to these spikes in temperature. And so they took the submersible vehicle, Alvin, and went down to this place, and they discovered a very rich environment full of life and clams and all kinds of life forms. And they took a lot of photos, of course, and they were astonished by what they found. And that was the time when you didn't have smartphones, so you had to develop the film from the photos. So when they were developing these films, waiting for several hours, and when they finally got these films, they could see that the data set with the temperature spike and the mini earthquake were occurring at the same time, where they also saw all these life forms, which means that these life were surrounding a place on the ocean floor where you had heat coming out and where you had a very active, geologically active place. And the crew was surprised and astonished because before this, they hadn't known that life could exist in such amounts in the ocean floor. And they thought that it was almost barren. So because of that, they didn't bring a biologist to the crew. So it took 2 more years before a biologist actually could go there and take samples from this place. And this first biologist to go to this place was Fred Grassel in 1979. And this was a very large, it was a revolutionary detection or discovery. So it revolutionized the entire idea of life on Earth, because life could suddenly be was shown to be habitable not only on the surface of a planet, but also below the surface of a planet, which means that this life was kind of detached from sunlight and from photosynthesis. And with the discovery of hydrothermal vents, chemosynthesis was also discovered as an actual, unity of life form. This chemosynthesis had been discovered before in the laboratory by a Russian researcher called Sergei Nikolaevich Vinogradsky, who had done these experiments in the lab, and he could see that some microbes could use sulfur, iron, and nitrogen to live and totally detached from photosynthetic life, or detached from photosynthesis. So, but it wasn't until the discovery of the hydrothermal vent systems in 1977 that chemosynthesis was established properly as a new type of life. And also with the discovery of hydrothermal systems, the ocean water chemistry was revolutionized. After that, also over 500 new species have been discovered in hydrothermal vent systems. And it also sparked new ideas of life outside Earth, because if you could imagine a world where life can exist in a world where the surface is inhabitable, but the subsurface is habitable, using just chemical energy for as a metabolism, then you suddenly expand your places where you can find life. You don't have to be in the habitable zone in order to find life any longer. So this started a lot of new ideas where to find life, but also new ideas of chemical reactors sustaining life, and chemical reactors that were prebiotic, so the prebiotic chemistry leading to life. So this was a really revolutionary discovery. So if you look at hydrothermal systems, what type of life do we see here? So if we look at terrestrial biosphere, most of the biomass is from plants and fungi, and in marine environments, most of the biomass consists of protists and animals. Whereas in the subsurface, deep subsurface, where hydrothermal vent systems are a big part, most of the biomass is from bacteria and archaea, which are unicellular organisms, and it's much harder to analyze these kind of organisms than plants and fungi, for example. This means that it's much harder to explore the subsurface and the hydrothermal vents than terrestrial environments, for example. But that also means that there's a lot more to discover. And some estimations have been done to how much biomass that is in the subsurface compared with the terrestrial environment. And as much as 41% of the biomass has been suggested to be in the subsurface environment. That is disputed, of course, because it's quite difficult to estimate the actual biomass. And it's especially hard to identify what organisms are living in the hydrothermal vent systems. And what are they living from, these chemosynthetic microorganisms? If we look at the hydrothermal vent system, this is a simplified image that I think is very telling, because you can see all the chemistry going on here, that you have a lot of seawater flowing through the rocks, dissolving the rocks and producing all these ions and gases and metals that are dissolved into the fluids and then flowing out from the hydrothermal vent system back into the ocean, where it's oxidized or somehow precipitating out, falling down to the ocean floor again. So you have the circulating water and with circulating metals in the ocean. And here in the dotted line, you have the microbial biosphere living within the rock, but they are also living in the plume here. But within the rock, where the temperature is moderate and where they still have possibility to access the fluids that are percolating through the rocks. But they are also dissolving the rocks themselves. So when people have tried to analyze what kind of microorganisms that are within these rocks, they find almost the same rocks as they find in terrestrial deep subsurface environments. So there are sulfur reducers, sulfur oxidizers, nitrogen reducers and oxidizers, and all kinds of chemical reducers and oxidizers. And in a system where you have a lot of serpentinization going on, there is a high production of hydrogen, so you have a lot of hydrogen oxidizers. But you also produce hydrogen in these environments from the microbes themselves. So you have a very vast number of possible metabolisms going on in this. And you have also environments that both are anaerobic and aerobic. And this is an image showing approximately how a chimney wall of a black smoker is looking like. So you have the vent fluids coming up here from the chimney, and you have these fluids are of very high temperature, and you have a temperature gradient going from 350 degrees down to 2+ degrees water in the ocean water. And you also have all kinds of gradients, nutrient gradients going from the vent fluids into the seawater, and you have all kinds of microenvironments that these microbes can live in, and gradients of pH values. So within a quite small area, you can have all kinds of gradients allowing for anoxic and oxic environments and anaerobic and aerobic microorganisms living very close to each other. So the hydrothermal vent systems are host to all kinds of life forms. But it's quite difficult to sample in hydrothermal or deep marine hydrothermal systems, at least, especially the living organisms. You have to either drill into the ocean floor or to go by submersibles, and that's a very expensive thing, and it's also quite a high risk of contamination, even though now they use something called the cork, which is it's a plug to stop the ocean water from entering the drill hole and contaminate the sampling. So it's the samples that are taken up with this device, the cork, is much less contaminated, but it's still a very expensive and difficult sampling when you're sampling living organisms from the deep subsurface. So one way of another way of doing it is to do some terrestrial sampling, to go to an environment where you have active serpentinization or an ophiolitic sequence, so a fossilized hydrothermal system. So I was going to tell you a little bit about what we have done in such an environment. We went to a place in Turkey where you have an active serpentinization environment where rainwater is flowing through cracks that come in contact with the ultramafic rocks in the deep and starts the serpentinization reaction that heats the temperature or heat up the fluids, and it's emitting gases, and the water is flowing out on the surface of the outcrop. And the waters are warm and with a high pH. So, but it's also an fossilized ophiolitic sequence. So we went there to both take active living samples to look at the microbial community structure, but we also took samples of the rocks to look at the fossilized evidence of microbial communities within the rocks. But what we found in the living community on and within the ophiolitic waters was thermophilic microorganisms, of course, because the temperature was quite high, up to 80 degrees Celsius at some parts. And we, of course, found a lot of hydrogenotrophic bacteria that lived from the hydrogen that was formed from the serpentinization reaction. And we found sulfur-reducing bacteria, metal-resistant bacteria, and nitrogen fixators. All kinds of bacteria that is also very common to found in marine hydrothermal vent systems. The only thing we couldn't really extract from the DNA was archaea, and especially methanogens that we were extra interested in, because methanogens use hydrogen and carbon dioxide as an energy and carbon source. We couldn't extract that due to the low cell density of the archaea. So what we did instead was that we brought bottles of methanogenic growth medium in sealed bottles, and we took some of the liquids that were flowing, or the water that was flowing on this outcrop, and injected it into the growth medium bottles. And we took them back to the lab and analyzed what was growing in these bottles. And we could then isolate one, especially one. We saw a lot of different methanogens, but one especially was very strongly growing, and that was a methanogen called Methanobacterium oryzae, and it's an obligate anaerobe. And we were quite surprised that anything grew because of the thin water layer that were flowing onto this outcrop, where air is flowing, and it's a really aerobic environment. So we were surprised that we got any methanogens at all growing in these waters. And we took samples not only from the waters, but also from the sediments underneath the water, and we found the same results. And this Methanobacterium oryzae is filamentous methanogen that is, it was growing like crazy. As you can see in this picture, it's full of filaments growing everywhere. And it was quite interesting because it was abundant almost only in the samples with a high iron content. And we think that this is an ongoing research, so it's still a little bit of a mystery, but we think that it forms these huge colonies that clump together, and that the surface of this, or the outer part of this colony, is affected by oxygen, and the inner parts are anaerobic, so that they can still live within these huge colonies, because it's not forming these biofilms that otherwise can protect from oxygen. So yeah, we think that they are forming these huge colonies. And then we sampled the rocks themselves, and we could see that these methanogens were also living inside the rocks and in all kinds of rocks from the serpentine environment. And most of these rocks, if we looked at the functional genes, they were using gases. So most of them were hydrogenotrophic. And some of these microorganisms also are biomineralizing, and we could find a lot of biominerals forming in these waters, and they form metal carbonates, iron oxides, and manganese oxides mostly. So I was going to talk a little bit about microorganisms that are living within rocks, so in the dark, using rocks to extract the nutrients they need. So microorganisms living in rocks, they can extract nutrients from the rock directly, or by consuming the already dissolved elements in the solution. So if we look in a hydrothermal vent system, we can have microorganisms living in the chimney attached to the rocks, dissolving the rocks and extracting the nutrients from the rock, or we can have those microorganisms that consume the already dissolved elements in the fluids or in the plume of the hydrothermal vents. So if they extract nutrients from the rock directly, they can alter the rock in three major ways. Either they extract acids to dissolve the rock, or they extract chelates like siderophores or they alter the redox chemistry of the rock. So they dissolve the rock by reducing or oxidizing the rock surface. And in this way, they will gain access to the nutrients. And if there is an excess of nutrients that makes the fluids toxic, they can handle that by biomineralizing, for example. And they can biomineralize carbonates or oxides or phosphates or sulfates. So they can kind of create their own balanced environment by extracting the nutrients they want and biomineralize the toxic excess metals into new minerals. And for that, they are, for the extraction of nutrients, they are using siderophores mostly. And that's an organic compound with the primary function to chelate ferric iron. And they are doing this to make it bioavailable for microorganisms. So, and here is an image of one siderophore. It's an organic compound that kind of surrounds the metal ion and binds it, and then makes this metal ion available to for the metabolism of the organism. And iron, most of the chelating compounds are specific to iron, since iron is the most essential element to organisms, and iron is used both in enzymatic processes and oxygen metabolism, electron transfer, and a lot of enzymes that is essential to the microbe. So, and here is an image showing how they can do it. Either they attach themselves directly to the rock, releasing the siderophores, extracting the iron directly, or they can release the siderophores, for example, in a plume, and the siderophores that are free-flowing will combine with the iron and making it available to the microorganism. And there are pros and cons to both ways. The pro for attaching directly to the rock is that they can control exactly how much iron they want and only extract the amount that they want. But they are attached to the rock, so they cannot move away from the rock that easily. Whereas in this microorganisms that are free-flowing, they can move to the source of nutrients. But on the other hand, the amount of iron is diluted in the solution, so it requires more effort to fetch or to gain the iron that they need. And if you look at the hydrothermal vent system, it's extremely rich in metals. So in many ways, the microbes in a hydrothermal vent do not need so much, perhaps, to extract nutrients from the rock. So instead, one of the ways that they need to handle this environment is through immobilize the toxic amount of metals in the hydrothermal vents. And they can either immobilize the toxic amounts of metals through biomineralization, or they can, as I told you before, extract the nutrients they need by chelation. And biomineralization is a microbial strategy to cope with this metal toxicity. So they induce biomineralization to get rid of the excess metals in the solution. And one pathway to biomineralize is through the nitrogen cycle, where urea, which is suggested to form in hydrothermal vent systems, is hydrolyzed by an enzyme called urease. And when this urease enzyme is hydrolyzing the urea, it creates an increased pH. localized pH on the surface of the cell. And this inevitably precipitates minerals, the pH rise, many minerals, such for example, carbonates. And so what they do is, here is an example of a mineral that is using urease to biomineralize. So they use the urea and they hydrolyze it with the urease, and they are producing ammonia and water and carbon dioxide. And this carbon dioxide is balanced in the cell with some other enzymes, and they are extracting hydroxide ions and bicarbonate ions. And these bicarbonate ions can combine with any excess cations floating around. Mostly it is calcium ions, and combined to form calcium carbonates. And this can happen on the surface of the cell, encapsulating the cells themselves, or it can happen in the solution. And some microorganisms can control this very specifically. So they can, for example, use this urease to build shells. Coccolithophores are one of those organisms, for example. And there are 4 factors controlling the biomineralization of carbonates, and that is the concentrations of cations, the dissolved inorganic carbon, the pH, and the nucleation sites. And biomineralizing microorganisms, they can control all these 4 factors themselves and control this environment by themselves by activating this urease, for example. And here are some images of carbonate that has precipitated outside of a cell like this. So by raising the local pH directly on the cell surface, making the carbonate precipitate directly onto the cell. And this, if there are microorganisms that cannot really control it, but they use urease for their nitrogen metabolism purely. And so this is a side effect, this raised local pH. And then this precipitation onto the cell surface can be deadly for them because it's encapsulating the entire cell and just killing it directly. So it's only a few microorganisms that control this biomineralization, and there are a few that do not benefit from it. But we wanted to look in some experiments if we could use this urease and urea reaction to not only biomineralize carbonates, but also oxides. Because if you raise pH by hydrolyzing urea with urease, you should also be able to precipitate oxides. If you look at this EH-pH diagram, for example, if you have manganese in the solution in a normal water with EH of 0.2, and you raise the pH, you will inevitably precipitate out oxides of manganese. So we wanted to look if we could use urease and urea to increase pH and thus oxidize manganese. And we could see that within 2 hours, we could increase the pH from 7 to 9 and precipitating either rhodochrosite, which is a manganese carbonate, or form manganese oxides, or both in the same experiment. So biomineralization of metal carbonates and metal oxides may form through this process. And since urease is an enzyme that is both intracellular and extracellular, that means that if we have a lot of microorganisms living in a hydrothermal vent system and they extract these urease, this urease will function by itself outside the cells to split urea, increase the pH, and precipitate out minerals within this system. Life has kind of adapted to life in the hydrothermal vent system, handling both high temperatures and all kinds of pH values, and making use of the chemical energy. But the microbes has also learned to adapt to high toxicity of metals by either having developed genes to be able to cope with high concentrations of metals, or by biomineralizing them. And all this is independent of the surface environment and its habitability. And this means that if we are going to look for life, we can look for life in other places in our solar system where the surface conditions are harsh, but has hydrothermal activity. And a few examples of such environments is Ganymede, Europa, and Callisto, which are all moons of Jupiter, where they have also water ice, and some of them have oxygen, and they have silicates, and in some cases they have also been suggested to have hydrothermal systems, which would then be able to possibly sustain life underneath the icy surface. And there is also, of course, the water plumes and Enceladus that is suggested to sustain life. And we hope that this will be the case and that we find life soon in either of these moons. And that was all I was going to talk about today.